Hey everyone, it's your host Russ Altman from The Future of Everything. Our team has been working hard on some new episodes, and we've got one more best of from the archives to share today before we bring you another new guest real soon. Not too long ago, I spoke with one of my bioengineering colleagues, Fan Yang, about some of the fascinating work she's doing in the area of tissue engineering. I hope you'll take another listen to hear more about the ways her lab is modeling human tissue to make sure we can develop better understanding for replacing damaged tissues that occur in many different diseases. Before we jump into this episode, I'd like to ask you to rate and review the podcast. It'll help others figure out if they're interested in the future of everything. Healing is a great ability that we have as living organisms. Skin, cut it, it heals up. Bones, break them, they get fixed. Even liver can regenerate. So there's quite a capability already built in for humans and other living organisms to regenerate damaged pieces. But some organs are less good at that. And in fact, after injury, they don't regenerate very well. This includes cartilage. There's a scourge of arthritis all over the world right now as the population ages. Knees, necks, backs, hips, they're all made out of cartilage and they're all starting to fail as you get older. Kidneys and even brains don't regenerate very well. So tissue engineering is a part of bioengineering that looks at how can we create new and improved tissues to put back into the body so that we can get that nice cartilage for pain-free walking, running, living life. Now, there's many challenges to doing this. The tissues that we grow in a dish don't really adopt the right structure. But even if we can grow them, we need them to have a blood supply. We need them to be the right shape. We need to figure out how to get them nutrients. And we need to figure out how to get them into the body. We could use surgery, but it would be even better if we could just inject them. The other thing is we'd like to take advantage of the body's natural ability to regenerate. And maybe we can help the body by just creating the situation where the cartilage, the bone, whatever, regenerates naturally. So you need the right 3D environment. You need the right mix of cells, not just heart cells in the heart. There's also connective tissue cells. There's neural cells, neurons. So you have to make sure that when you're regenerating an organ, you have all the right cells. You need to make sure that the environment has the right stiffness. Brains are mushy. Bone is very hard. Brain cells are expecting to be in a mushy environment. Bone cells are expecting to be in a stiff environment, and that matters. Then, of course, blood supply and nutrients. Well, Professor Fan Yang is a professor of orthopedics and bioengineering at Stanford University. She creates new methods to create replacement tissues and systems for studying how diseases arise and maybe how we can generate new therapies based on more accurate modeling of the environment in which a tissue develops, grows, and lives. Fan, let's start out with just a question, pretty basic. What is tissue engineering or tissue regeneration, and why should we care about it?
The tissue engineering field really emerged as a solution to address a very critical unmet medical need, which is, when people, have, for example, late stage arthritis, right? They will need to have another way to get a healthy joint back.
Right, because their cartilage, for example, is just a mess.
Yeah, or for people who have heart diseases, they may need a new heart valve or maybe other muscles. So tissue engineering really emerged as an effort to try to grow this kind of biological tissues in the lab so that people would have some biological viable tissue that is very similar to what they have lost to start with. That is not based on metal, that is not based on plastic, which is actually what people are getting as they get total joint replacement these days. So the idea is to go from just restoring the mechanical functions or electrical functions, but to really, can we restore the biological function with material structures and your own cells that are similar to what you have lost?
And it seems like a huge benefit of that would be presumably these might last longer and be much more natural for the patient instead of having a piece of metal inside or a piece of plastic or a piece of another animal. As far as I know, we don't routinely use engineered tissue in patients. I'm sure there are some exceptions, but what are the major technical challenges as you see them to getting really good tissues that we can give back to patients who have damaged tissue?
Yeah, in fact, I would say as this field evolves, we have made tons of progress in the research arena. For example, now people can actually use stem cells or patient's own cells in combination with what we call biomaterials to actually grow a beautiful piece of skeletal muscle. Or, in my lab, we are very passionate about growing a piece of cartilage, bone, etc., right? So that actually has become a reality in the lab. The challenge, though, is that you can make a beautiful piece of tissue, but then you have to figure out how to put that into the human body and still keep it viable, but also integrate with the rest of the body. I think that's where the challenge is, that, when you grow something in the lab, you can give them all the oxygen, all the media, their favorite food. But once you put them into the body, in order for them to survive, they have to first, for example, the first challenge is how do they integrate with the local blood vessel supply so that they can stay viable? And the second issue is that how do we also, eliminate or at least avoid unnecessary attacks from either local immune cells or other cells so that this tissue will actually continue to be viable. So I would say that has been a huge challenge, is to scale it up to the size that is relevant to human, because a lot of work we do in the lab are in animals, right? And humans are, substantially bigger when you think about the size of anything.
Right.
And vascularization of a sizable tissue is a challenge. But also, I would say another challenge is just to also recapitulate the complexity. Because when you think about repairing one tissue, we're actually not talking about one single cell type. There are multiple cell types that are, in a play. So I think that those are all some of the realistic challenges when you think about such a complex biological product.
Yes.
And because of that, I think it has made it quite slow when it comes to jumping from the research to actually clinical viable product. Is how do we figure out about manufacture this at a clinical grade and also validate that this will actually scalable to human, which means clinical trials. And that's a very long and very expensive process. Depends on how complex your solution is.
So I loved your description of the and it made perfect sense that you can grow it in a test tube or a dish, but when you put it in a person, you have to make sure it gets blood supply. You're going to sew the person back up. And so you have to make sure that tissue is happy and doing well. And it's really great to hear that you're already able to kind of grow some of these tissues. I did want to ask, however, about the shape issue. So like when I have my knee, I need my cartilage to be a certain shape, but you can't just put a flat sheet of paper or of cartilage, but it needs to like mold to my knee. So how are we with respect to the three-dimensional aspects of tissue engineering?
Yeah, I would say that's probably one of the easiest problems to solve.
Oh, good.
Because there are multiple technologies available. First of all, like in the old days, you may have to like make something and the surgeons would have to manually cut and fit, right? Which is still very labor intensive and can be challenging. But nowadays we have a lot of advances in 3D printing. So, people can easily actually 3D print whatever shape because they can scan the patients to know exactly the shape, whether it's for bone, whether it's for cartilage, whether it's for anything else. The other thing also, the good news is that we also have better materials that allow us to actually not necessarily just make something outside and then put in to fit, but rather we can actually inject them in situ. So whatever shape you have, doesn't matter. We can inject it, fill exactly whatever shape you have, and then polymerize them in situ. So polymerize is a word we use a lot in biomaterials, meaning that they go from an injectable liquid phase to a solid phase. So if we do what we call in situ polymerization, it's actually no longer dependent on the shape, because whatever shape you have,
it'll just adapt to whatever it
exactly
sees.
One analogy to think about is that, for example, if you think about people who do house remodeling, if they have a big hole in the wall, right? If you have the right patty, you just put it in and you fill it up, and then once it's dry, doesn't matter what shape is your hole, it fills in perfectly. So that's actually one thing we have been developing in the lab, is to make something that is injectable, but also can actually function like a patty so it doesn't flow away. And you can then let it turn into the solid phase whenever we're ready.
You mentioned cartilage as one of your passions. And I know it's a huge issue with the aging population. Many, many people are getting arthritis. It is very disabling to have a bad joint. Just to think about cartilage and to make it real specific, how good is the cartilage that we're growing, and how are we doing at getting it to grow blood vessels? And do we have all the right cells? You said that this is complex. It's often not just one kind of cell. So can you give us a little bit of a deep dive into how's the cartilage problem coming along?
I would say cartilage is actually a unique tissue. In one sense, that was the first target the tissue engineering community started to tackle because it was thought to be an easy problem. Why? Cartilage does not have blood vessel. It's very rare. If you think about, when you go to a grocery store and get your chickens or, pork, you probably see this white shining thing on the surface.
Like at the end of the chicken leg.
Exactly. And that's cartilage. And why is it white? Because it does not have blood vessel. So people started with cartilage thinking this would be easier because we don't have to worry about blood vessel supply. And another thing is that cartilage is relatively simple in the sense that it also doesn't have, neurons and nerves and all that stuff. So it's a relatively simple tissue. But on the other hand, it's actually turned out to be one of the hardest problems to solve because cartilage is actually very dense in matrix and very, very few cells are actually viable in the end once the cartilage is fully developed. So that's actually why when people lose cartilage due to sport or due to trauma, it's really a one-way street.
In the sense that it doesn't grow back.
Like, for example, if you think about kids, right, they have bone fractures all the time, but typically they don't need tissue engineering. Why? Because bone actually is very well vascularized. There's a lot of blood vessels, there's direct access to bone marrow and a lot of stem cells. But cartilage actually doesn't have those access. So in a way, I would say where we are for cartilage right now is that we can actually make beautiful cartilage in the lab. In fact, we can also get pretty reasonable cartilage regeneration in the animals. The challenge is that when you go into the human, though, there are issues with, scalability. The other thing is also just compared to what is currently available for the surgical intervention, which is actually a standard of care procedures called microfracture. What the clinicians do is that they drill a hole through actually the cartilage into the bone layer that is underneath. And the reason they do that is to bring up the stem cells from the bone marrow, hoping that, and also the blood, which contains all of the different growth factors. So hoping that that would help with the cartilage regeneration. And it actually does help the defect to be refilled to some extent. But the problem is that it's a scar formation in cartilage. So what we have is actually typically what we call fibrocartilage.
So it's not the smooth, beautiful cartilage that would help your knee move, but it's a little bumpy or not really fit for purpose.
It's not about smoothness, but it's the composition. And in tissue regeneration, we know structure and mechanical function are closely related. Even if the space is filled up to a very smooth level, if the composition is not right, then you don't have the right shock absorbing function, which is key for articular cartilage. So I would say with regards to the research side, the other challenge is the speed of regeneration. Because a lot of the products developed for cartilage, for example, are injectable. And because exactly the concern you had is that those defects usually come in very weird shape, right? So injectability is very desirable for a couple of reasons. Number one, it will be allowing the surgeon to actually use injection, which is a procedure they're already very well trained for. Number two, patient likes it because it's minimally invasive, right? You can avoid some big open surgery. Now, so what we have been working on, though, is to try to actually have injectable materials that also can accelerate cartilage regeneration in a way that not only that we're thinking about stem cells, but we're actually thinking about also the other supporting cells in the defect. Because inflammation, for example, is always there, right? And how can we then, allow the healthy dose of inflammation initially, but also prevent the undesirable long-term inflammation, which will actually prevent the tissue regeneration. So that's one thing. And from that, towards that goal, for example, one platform that technology our lab actually invented for that purpose is something called microribbon-based hydrogels.
Great. I wanted to ask you about it because it's such a great name, but it's not clear what it is. So please tell me about microribbons and why I should be excited about microribbons if I have bad cartilage.
Okay, sure. Well, so basically hydrogel is a kind of biomaterial that is used a lot in medical application and also in non-medical. The definition of a hydrogel is that it's a polymer that you can dissolve in solution. So it's a liquid phase. And then upon a stimulus, which can be light or some sort of chemistry, then they will go from a liquid phase to a solid phase. The unique thing about hydrogel as a biomaterial is that it has very high water content. In fact, there are a lot of hydrogels you're already familiar with in your everyday life without maybe realizing. Your contact lenses. And actually diaper is a super absorbent hydrogel, tons of water without, breaking apart.
Okay, grateful. So these hydrogels are like these things that are similar to, and it says gel and it says hydro like water. So we can kind of imagine, especially with the diaper analogy and the contact lens analogy. So now going back to the micro ribbons, they're a special type of hydrogel?
It's a special shape of hydrogel that we have made for the purpose, actually, for regeneration. And in fact, initially motivated for cartilage regeneration. So we love hydrogels because it's injectable, right? But on the other hand, we really struggle, not just us. I mean, the community also kind of struggle with hydrogels for a couple of reasons. Number one is actually the size mismatch. So if you think about a hydrogel, I just described that in order to form hydrogel, it's basically polymers in a solution that then polymerize and become a network. And that's how they become solid.
Yeah.
But then what is the pore size here? We're talking about basically the size of the porosity in hydrogel is basically the distance between the two closest cross-link. And those are on the order of a few nanometers. That's the typical polymeric network, pore size, right?
What you said pore, like these are the size of the holes so that things can get in and out.
So now that if we use this for cell delivery, so literally the cells are being what we call encapsulated or suspended in the hydrogel and then become a tissue, right? The problem, though, is the size mismatch. So cells, they're not nanostructures. They're actually on the order of microns. Typical cells, we're talking about 10 micron plus minus, depending on the cell type.
So they're much bigger than a nano.
So when we put the cells in a hydrogel, on one hand, it's awesome, right? Easy to mix. You can deliver it, minimally invasive. The problem now, though, is that cells are becoming this kind of micron-sized monsters being present in a nanocage. So in order for them to do anything good, whether it's proliferation, migration, or producing new tissues, the first thing they have to do is to basically break down whatever the physical constraints that is imposed by this network. So the microribbon actually was designed to overcome that. So what we thought about is that, well, instead of just making hydrogel directly into a three-dimensional structure for cell encapsulation, what if we just make them first into a building block? And then this way we can mix them with cells. So the cells would actually be on top of the ribbons rather than being physically entrapped within a hydrogel network. So the vision here is that we want to have a three-dimensional highway system for the cells. Imagine the cells to be like the cars, right? Doesn't matter what kind of defect you have. Could be cartilage, could be heart, could be blood vessels, right? Literally, when you have a wound site, you have a cavity. And when we put our biomaterials, in this case, microribbons, into the cavity, what we are actually providing is basically a three-dimensional, highly interconnected highway system. And then now the cells from the surrounding tissues can easily drive in. And this is actually where it is very desirable.
So if I'm understanding, the old kind of hydrogel didn't give enough elbow room for the cells to move around, to divide and expand. The fettuccine, the fettuccine that you've created creates still a 3D supportive environment, but allows the cells to move around, to go into new places, and that just will facilitate the growth of the tissue in the ways that you want it. Is that in the rough ballpark?
Yes, you have summarized it pretty well. And I would say one thing to also think about here is that not only just the cells we put in, but in this case, as we think about clinical translation, we actually want to put in these microribbons into the human without having to use cells. Because the idea here is that once we put in the right material with the right composition, we can actually engage the body because our body already has tons of good cells, whether it's stem cells, vascular cells, or other immune cells, right? The question, though, is that can we get the right cells to come in at the right time so that they actually collaborate with each other in a synergistic manner? That's the key. And the reason we also want to go with that is because from the regulatory perspective, if you have to take cells out from a patient, do something in the lab, and then put it back, it's actually a very high bar for regulatory, and it takes a lot more money.
When you say regulatory, you mean like the FDA?
Exactly. So if we could actually use a simpler formulation, in this case, just the biomaterials without additional cells that we add onto it, right? It's actually a much simpler product to manufacture for quality control. It's also cheaper for the patients.
Your work also includes creating tissues, not so much for implanting into people, but to study disease. How do we use these technologies not to create therapeutic tissues, but to understand disease and maybe even help with drug discovery and things like that?
Yeah, great question, Russ. So in my lab, over the years, we have developed these beautiful different biomaterials tools that are very good at making stem cell to become bone, to become cartilage, muscle, tendon, etc. Right? But then one thing we also realize is that this is the same environment that cancer cells arise from. And there's a huge need in basically, better understanding how cancer cells actually develop in our body, and also to be able to have, experimental models that is not in an animal, not in a human, that allow us to do drug discovery, because that would be a huge, benefit for a lot of patients, right?
And this might all be done outside of people, animals. It would be in a dish.
Exactly, but in a fancier version, because typically when you think about growing cell in a dish, they're basically being plated into some sort of plastic surface where the cells will just adhere and spread and etc. Right? That's actually the current standard when it comes to do cancer cell therapeutic screening. But one thing that researchers have realized is that the cell signaling highly depends not just on the genetics within the cell, but actually on the environment that they see. So for example, brain is a very soft tissue. We're talking about a stiffness of anywhere between 40 Pascal to maybe 1,000 Pascal.
No, I don't know what that means.
Give you a reference, right? So we're talking about 10 to the third here, right? At most. But when we think about plastic, we're talking about 10 to the 9.
So plastics are way stiffer than brain.
So because of that, when you put a cell into a very different environment, a lot of signaling already changes. And also when you grow cells in such simple environment, we really cannot study how they invade, or which cancer metastasis is a big problem, right? So that was the motivation, is that we say, why don't we leverage on all these beautiful tools that we have already developed, initially for mimicking the normal tissue development, but in this case, twist it a little bit to use it to study abnormal tissue development in the case of cancer. So this is where I would say that really motivates us, is that we already have the tools, right? All we have to do is now to just adjust the target, the biological problem. The other thing I think is also very powerful that we have learned through this cancer modeling work is one of the biggest challenge in cancer therapy is actually drug resistance, as well as the heterogeneity. Right? So even when it comes to the same type of cancer, different patients would have very different genetic and epigenetic background, which means that they will not always respond similarly to similar kind of drugs.
Right. And we see that all the time, where some drug is a miracle for one patient and it doesn't work at all for another.
Exactly. But how do you test that, right? I mean, it's very difficult to test. Take the bone cancer as an example, right? So right now we're actually collaborating with another lab at UCSF where they have spent decades establishing all these patient-derived cell lines that are very well characterized using all the cool high-dimensional sequencing technique. But they have been relying on two-dimensional cell culture to do the drug screening. But what we have found now through this really great synergy is that if we put the same kind of patient cells into our 3D scaffolds that actually mimics the bone niche, their signaling is very different than 2D. In fact, a lot of the signalings from our characterization is lost in 2D, but is retained in our 3D culture. And then we can use this to our advantage in many ways. Number one is that now we can actually use this 3D model, and integrate that with newer techniques such as sequencing to identify what is actually retained in 3D culture, but is lost in 2D. So now we can then find drugs that specifically target those genes or those pathways, because that means now we can actually find drugs that has been missed out.
So just to summarize what I'm hearing is, you took the patient cells, and that seemed like a great thing because then you'll be able to do very specific. But when you put them on a plate or in an environment that doesn't really match what they're like when they're growing inside the human body, then they start acting funny. And this 3D model that you have created for the environment gets them to act a little bit more like they will be acting in the patient. And therefore, it's a much better readout for susceptibility to drugs and other measurements.
Exactly.
Which cancers are you focusing on?
So right now we're actually focusing on, for the bone cancer, we're focusing on osteosarcoma. It's a disease that impacts mostly adolescents. And the treatment has not improved over 30 years. So one thing that we are actually using this model for right now is to exactly address the precision medicine issue. So we're looking at different cell lines with different genetic backgrounds so that we can actually find, druggable targets that would be a lot more specific to do the right, cytotoxic killing, but without having the undesirable side effects. So I think that's something that we really hope to provide some very powerful tools that allow us to, find drugs much faster but also, with reduced cost and also save some animals.
So in the last 2 minutes, I did want to ask about how does this actually get into the hands of doctors and their patients? And I'm sure there are many, many challenges and barriers, but can you paint a picture for us about what's standing between the basic science that you're doing that looks like it's really going great and being able to really impact the patients' lives?
Well, now we're talking about product development, also
Yes.
regulatory approval process, which we kind of touched upon a little bit earlier. And then finally, it's also just commercialization process itself. I would say, if you think about just, in terms of the sequence, I think the first challenge is to actually, a lot of this work will have to be done in an industry setting, either through startups or through partnership with large, companies, right? To do all the manufacture that academia cannot do. That's one thing. But also, I think a lot of these techniques are very new. So FDA is actually also doing their best trying to learn what would be the right questions to ask. Because if they ask too little,
Right.
it's not safe for the patients. But if they're asking too much, then they're essentially slowing down the translation and approval process and
Right.
delaying the benefit for the patients.
I mean, they've been making drug approvals for decades, but they have not been doing this for decades. And so they really need to catch up with all of the new innovations.
Yes, absolutely. And I think also when it comes to in the end is that there's also this, I would say, transition phase for adoption of new techniques, right? Like early adopters will probably need to be physicians who actually understand the technological advantage so that they will then prescribe that for their patients for their benefit.
Thanks for tuning into this episode. We have more than 250 episodes in our archive, so you have instant access to a broad range of discussions on an amazing variety of topics. If you're enjoying the show, or if it's helped you in any way, please consider rating and reviewing it. That'll help us understand what you're experiencing with the show, and it'll help get the word out. You can connect with me on X or Twitter at RB Altman, and you can connect with Stanford Engineering at Stanford ENG.